I'm going to talk about primer on parasites, or basic of parasites, focusing on important internal parasites for small ruminant producers to know. What's the significance? Why it's important? Because internal parasites are the number one health problem of small ruminants, especially goats, in the southern United States, as well as temperate regions on the globe. Warm, moist weather is conducive to parasites. On the other hand, hot, dry summers are not a big problem because those conditions are hard on parasites. There are some important parasites for small ruminants. I'm going to show you two big categories of parasites. One is gastrointestinal nematodes, often abbreviated as GIN, that include Barbapol Worm, Hemoncos contortus is the Latin name, brown stomach worm, black skull worm, threadneck intestinal worm, Nematogelos. Those are gene categories. And another big problem is Coccidia. Probably many people know this Coccidia. It's problematic, especially for young animals. Among these important parasites, Barbapol worm is the most threatening parasite. It's been a problem in the temperate region on the globe. Let's see what is Barbapol worm. It is also called wire worm. The size of mature female is 0.4 to 1.2 inches or 1.0 to 3 centimeters. They are found in abomasum, the fourth stomach chamber, and one female could suck 0.05 to 0.25 milliliters of blood each day to produce 5,000 to 10,000 eggs per day. 5,000 to 10,000 eggs per day from one female. Can you believe that? This is a picture showing whole worm. As you can see, there are white part and red part twisting together. That is named for Barbapol. So I will explain with this drawing. This red part is intestine with full of blood, and this white part is oviduct with full of eggs. 5,000 eggs or more. It sounds crazy. Let's close up. This is magnified, 10,000 times magnified face of the Barbapol worm, a Barbapol worm. This sharp mouth poke into the wall of abomasum and suck the blood. It's kind of scary. Then this slide shows about infected abomasum of a goat. We can those small tiny thread shape thing is all Barbapol worm. I would say, let's say this has 3,000 to 5,000 Barbapol in the abomasum. So let's say 5,000 Barbapol worms can suck 1 cup of blood per day. 1 cup of blood per day. about 10% of the goat's blood. Okay, if you go to donate your blood, 200 cc of blood, you will be told, do not come back for 2 months, right? Then, now, goats are sucked a cup of blood every day, so you can expect what's going to happen. Here are symptoms of hemoncosis. Their nutrition is sucked with the blood, so of course they lose weight. They are sucked blood as well as their nutrition, right? So they lose weight, or of course they can get anemia. Then eventually they can die. If acute infection and they multiply so quickly, since the worms can multiply so quickly, then they can get acute death. It's really, really scary, threatening problem. But before being scared, let's learn about the worm. Here is the life cycle of Barbapol worm. I said Barbapol worm, but generally all gene group has same life cycle. So let's begin with the goats. Goats eat grass. Worms stay on the grass, so goats doesn't remove them. So goats eat grass with worms, right? Then worms get into the body of the goat. The Barbapol worms go to abomasum and start developing, and within 3 weeks they start laying eggs. Those eggs are excreted with head feces and drop on the ground. The eggs can hatch within 3 to 7 days when the temperature is warm enough, let's say 60 degree Fahrenheit or 15 Celsius, and humid. Then those hatched egg is already worms, right? Those larvae. The stage, they called it stage 1 larva, or L1 larva. L1 larva feeds on bacteria in feces, so they are still in the feces. Then after molt, they becomes L2, stage 2 larva. Then after they shed their cuticles protecting their body is shed, then becomes stage 3 larva or L3 larva. This is the infectious stage. And this stage, they are eaten by small ruminants. But important thing to know is this L3 infectious stage of larva or larvae, they cannot move a lot. They can wiggle, but they cannot travel. So basically they stay on the moisture, stay in the morning dew or raindrops. So to cope with this worm, we need to cut this cycle somewhere, right? Then we can stop the problem. As I said, goats eat the worms with grass, right? Especially these goats are eating very low height grass. As I said, worms cannot travel or stay in the dew, stay on the raindrops, right? So if the height of grass is this low, then goats get more chance to eat those worms. But if that grass is taller, then goats has less chance to eat the worms. And also, if you graze them, if you pasture the goats, put the goats on the pastures early in the morning when the morning dew is still there, then goats will have more chance to get worms, eat worms. But if you graze them, you graze animals after morning dews dry out, then also goats have less chance to eat the worms, get infected. So this is one cutting cycle. So like dewormers or other stuff is trying to kill worms inside the body of goats, right? But this if you know the life cycle of worms, then you know how to cope with them, right? Very important. So let's review about third stage larvae, L3. This is the infectious stage. Eggs can develop to L3 larva in 3 to 7 days. They prefer warm temperature, more than 75 degrees or Fahrenheit or 25 degrees Celsius, and humid. They have like special ability to be arrested in abomasum in winter. So what that means, they can hibernate during the winter when the environment is not suitable for them, like less than 75 degrees or in colder or dry. So this parasite problem is less during the hot, dry summer and winter. This is a picture of L3 infectious stage of larva in a dewdrop. You can see lots of larvae in the dewdrop. So you can imagine, that they are on the grass. Goats cannot avoid it to eat it, right? So this is how goats get infected. So I mentioned a little bit about the temperature but when is a problem? The problem is midsummer to fall. But not all worms like same conditions. For example, brown stomach worm like 60 to 85 degree Fahrenheit or 15 to 29 degrees Celsius, a little bit lower temperature. Black skull worm likes 72 to 92 degree Fahrenheit or 22 to 33 degrees Celsius. Barbapol worm like more higher temperature, 75 to 99 Fahrenheit or 24 to 37 degrees in Celsius. Not only the temperature, but there is also impact for animals. When the animals are under stress, then they have more problem. For example, lambing or killing season or lactating, those unused, those animals are under stress. They are producing they're consuming lots of energy to labor and production of milk, right? And also for kids, young animals, kids and lambs, weaning period is huge stress. Separating with moms, they need to change the feed, right? So those animals, moms, just delivered moms and lactating moms, weaning kids, those are more problematic. So it's happening especially during the early spring and midsummer. But why so problem, right? If it's internal parasite, why we cannot deworm, right? That's a big problem, because an anthelmintic or dewormer may not work because, number one, drenching failure, that's involving human error. As you can see the picture, when you deworm them, you want to drench them, right? You drench them, but all the dose may not go to in goat's body or animal's body. Goats can spit out, or sometimes drench gun is not measured well, or it's not calibrated. Some different reasons drenching failure can happen. Number two is reinfection after treatment. So after drenching, after deworming, if they stay on the pasture, the same pasture where it's contaminated with worms for a long time, they may eat the worms again, then they're infected, right? But at the same time, we do not recommend to move animal right after deworming. We will talk about it later when Dr. Steve will explain a little bit more about it. But reinfection is another problem. Number three is the biggest problem: dewormer resistance of worms. What dewormer resistance? The worms are resistant to dewormer. How? Right? Okay, this table shows dewormer resistance in the South Central US in 2017. We conducted a fecal egg reduction test at the farms in 5 farms in Kansas, Oklahoma, Arkansas, 2 farms from Missouri, and tested 3 different classes of anthelmintics. One was Barbapol from albendazole class, Eprinex, eprimectin, prohibit from levamisole. Numbers showing here are the fecal egg count. I will talk about the fecal egg count later, but briefly, the number of eggs in 1 gram of feces. So those numbers are fecal egg count before and after anthelmintic treatment. 1 time of treatment. We took the sample before the treatment or at the time of treatment, and after 10 days we took another fecal samples and conducted fecal egg count. The numbers in parentheses, red and green, those are the effectiveness of the dewormer. So in this fecal egg reduction test, if the effectiveness is less than 95%, then we call that farm has dewormer resistance. So for the group of Barbacin, 4 farms out of 5 developed dewormer resistance to albendazole. Only 1 farm in Missouri had 99%, very high effectiveness. This farm managed really well, control parasite very well, and they do the different measures to cope with this problem. The second class, Eprinex, they have all red. So by the way, this Eprinex or eprimectin is the same category, same class of ivermectin. So it's ivermectin. Everybody knows ivermectin or Ivomec, right? So Ivomec has long history of used in many different places in the world. So it may be not surprising, but it's not really working well. Third class, lebamizole. 3 farms out of 5 worked really well. They didn't have dewormer resistance, but 2 farms yet they had resistance to this prohibit. So what this really shows is that, for example, 85% of effectiveness of a dewormer, okay, so 85%. If a goat had 100 worms in the abomasum, you deworm him with a dewormer, 85% effectiveness, 85 worms got killed, 15 worms survived, right? So these 15 worms, each 15 female can lay 5,000 to 10,000 eggs per day. Remember? Each worm. Okay, so those survived was not killed by the dewormer, have resistance to that dewormer, right? So those female can produce resistant eggs, right? So this is how fast they can acquire the resistance to the dewormer. So every time you deworm them animal, the worms can have chance to develop resistance. See? So this is our message is please stop right now. Blanket treatment, scheduled treatment, drug overuse, misuse, underdosing. These are all helping to develop resistant worms. So please reconsider it, then stop it. Let's review what was the Barbapol worm. Adult worms, 0.4 to 1.2 inches, attached to walls of the abomasum, right? Egg size was small, like 550 micron. So this was Barbapol worm. other worms. Brown stomach worm. The egg size is the same, almost the same. Adult worms has 0.3 to 0.5 inches long, a little bit smaller than Homuncus or Barbapol Worm. He feed in mucus of abomasum. So this stays abomasum as well. And this worm causes diarrhea, loss of blood plasma, reduced appetite, poor performance. So they are not blood sucking. They feed on mucus. So they are nutrient sucker instead of blood sucker. Bankrupt worm is the size of 0.25 to 0.33 inches long, and they stay in small intestine. The size of egg is almost the same with Homuncus, Barbapol Worm. This one also causes diarrhea, reduced appetite, poor performance. Long-necked bankrupt worm has 0.4 to 0.9 inches long, feed in the small intestine, but this nematodulous eggs is much bigger than other gene worms, so you may be we can identify them. So this causes diarrhea, reduced appetite, poor performance. Nodular worm is 0.5 to 0.8 inches long and stays in small and large intestine. This also causes diarrhea, weight loss, hypoproteinemia. Whipworm. It's much longer, 1.4 to 2.75 inches long, and feed in the cecum and colon. So, but these are also blood suckers, causes blood loss, contribute for blood losses, and poor performances. So those gene groups, different. I introduced different types of gene groups, but in natural settings, it's very rare to be infected on one species of gene worm. Normally, if animals are infected, then multiple species we can find in feces. But Homuncus is the best multiplier, so usually dominant is the Homuncus. Of course, different environment, different temperature has little bit more different populations of worms, but like in temperate regions or warm regions, the Homuncus is normally the dominant species. Other internal parasites. Tapeworms. They are huge or very long, as you can see, up to 6 feet. So when they comes out with poop or feces, you can really see this tapeworm. This is causes slow growth, poor performance, but you can kill him with Barbacin. Control him with the Barbacin. And Coccidia, I mentioned a little bit earlier. This causes diarrhea in stressed animals, especially weaned kids and or lambs and transported animals. You can prevent by feeding medicated feed like Lumensin or Decox in the United States. And you can also treat with Albon or corrid. So these parasites, this Coccidia, is feces to oral split. So whenever you feed animals, please clean the trough or water. Goats and sheep can poop in the trough, not purposefully probably, but they can poop, like if those troughs are contaminated with poops, then it easily transfer to the other animals. So please, whenever you feed them, just clean the trough and water. That's important to control Coccidia. Lungworms. This lungworm causes chronic cough, and it cycles through snail. So for this problem, you better get veterinary assistance to this problem. And meningeal worm causes progressive paralysis of tail and rear legs. Excuse me. And this cycles through deer and snail as well. So you want to get veterinary assistance on this problem too. So this meningeal worm infects those nerves, so it causes problem. So I showed you different types of parasites and problems. So now we need to learn how to control these parasites in general, and also specifically for the gymworms or Haemonchus, Barbapol Worm. In general, number one is keep your animals healthy. Good nutrition supports good immune response. Right? We learned from COVID experience, if you are good nutrition, you're good health condition, then you will be able to cope with the problem, COVID. But those people who are old and less health conditions, then, or immunocompromised, then those people are weak to the COVID, right? So it's the same thing. Good nutrition, good health supports good immune response. Then they can fight with the diseases. So to make immune cells, they need protein. Protein is a source of immune cells. So protein supplement is important, especially for animals grazed on the low protein grass pasture. Keep in mind. Number two, genetic improvement. If genetic improvement sounds difficult, right? Like you have to keep the record, you have to analyze animals, like lots of things. You have to calculate many things. No, just keep healthy animals. Cull weak animals. That's the easiest way to, but it's still, maybe it's not as fast as calculation or recording, but still it works. So keep healthy animals and cull weak animals. Well, when you think about genetic improvement, there are two categories or two ways people talking about. One is resistance and the other is resilience. What the resistance mean is, resistance is an ability to control worm population in the body, animal's body, via immune response. So, like, what that means is when animals are infected, they somehow in the body, they can control worm population. They can somehow, they have like the immune response. With their immune response, they can control those multiplying worms from multiplying worms. On the other hand, resilience is an ability to maintain health conditions when infected. What that means is that animals that are infected, they produce, the worms are multiplying, worms laying lots of eggs, but somehow they can keep their conditions, keep away from becoming anemia or lose appetite or lose body condition scores. So which is you want to choose, right? We always want to choose resistance, because resilience animals still contaminating pasture. They're producing lots of eggs. Not goats are producing, but the worms in that goats, resilience goats, produce lots of eggs and contaminate pasture. So those contaminate pasture, the pasture is contaminated, highly contaminated, and more animals get chance to become sick, infected, right? So we always want to choose resistant animals. Number 3 is non-drug alternatives. Non-drug alternatives include co-grazing. Co-grazing does not mean sheep and goats together. Those are both small ruminants and they share the worms, so it doesn't work. But co-grazing small ruminants and cattle or horses works. How this does is cows, horses, they can eat worms, but they will not get infected. So if you graze them together, then cows, horses eats up those worms, then without being infected, they clean the pasture. So one alternative. And second alternative is pasture rotation if you have luxury of good amount of pasture, right? So pasture rotation is you divide your pasture into many, then put the animals in the pasture, then rotate. But this rotating duration is, if the duration is less than 30 days, then it may not work. Some study shows that. So at least recommendation is at least 2 to 3 months opening and between the rotation intervals. So but you can, during those 2 to 3 months, you can put cows or horses instead of opening the pasture or free from animals. So what I mean is if you put the goats or sheep in the pasture, after remove them, you can put cows or horses another 2 to 3 months. Then it works. You can still utilize that pasture. The next one is condensed tannin. This is a big research interest, being a big research interest to cope with this parasite. But one promising plant is Sericea lespedeza. The Sericea lespedeza is one of the common legume in South US. South Central US, but those animals who eat Lespedeza will not get highly infected. So this, especially those, if you feed Lespedeza hay or Lespedeza, then study shows the maturity of worms or the production of eggs of the worms are reduced. So it works. Copper oxide warrior particles. These copper oxide warrior particles are used to supplement for animals who are copper deficiency, but some study shows this has also effectiveness of reducing the Barbapol worm, not other worms, but Barbapol worm. The last one is nematode trapping fungi. It's more recent product, but Bubba is available in market now in the US. And the cool thing is this fungi trap the worms, like just you can see in the picture, then feed up this worm to produce spore. So it's kind of cool system. Then this Bubba does not develop antibiotic resistance. It's totally different way. So this is a very interesting and new strategy. But we don't know yet really clearly how effectiveness is it. And also it's still very pricey. So it's still developing, I would say. Number 4 is targeted selective treatment, TST. TST is only deworm animals when needed. For example, as this picture shows, if you have 18 goats, then only 3 are less healthy, then you treat only them, not all of them. This strategy keeps refugia. We will explain refugia later, but briefly, the worms who are not, those are not exposed to the dewormer, so susceptible to a dewormer. So TST slows development of antibiotic resistance in worms. So this is very, very important strategy. Number 5 is integrated approaches. Remember, keep animals healthy, genetic improvement, non-drug alternatives, targeted selective treatment. Those control the measures, strategies I explained is each important, but none of them works 100%. So we need to integrate all these approaches as much as you can to control the problems, this worm problem. Let me introduce some example. In a example, I have a friend of a goat producer. My friend's uncle started goat raising like 3 years ago. Then now he has about 40 animals. I visited him, my friend's uncle, a couple months ago. Then he is keeping 40 animals in the backyard. and like they are beautiful and healthy. No, I didn't see any score. Everybody's happy like that. Very shiny. Then, but the uncle's question to me was, do I need to deworm him? I said, no. Why? Then he said his friend's goat producer told him, if you keep goats, then you have to deworm them periodically. Then I said, no, don't listen to him. All the animals are healthy and they don't have any problem. Why you do need to deworm them, right? Only if one animal or two or animals who are less healthy and the problem you find, then at that moment you can think about it. But normally those healthy conditions, worms are not problem. So if the condition he keeps animals very healthy, that is good strategy to prevent this problem. So, okay, I talked about targeted selective treatment. Now, the question is how you can select those animals who might be infected, right? So how do you know? Now, diagnosis of worm infection. Number 1 is fecal egg count. So this measure counts the number of eggs in 1 gram of feces, 1 gram of poop, right? It's different from presence or absence of eggs. It's a number. Then we measure the severeness of infection. This is the most common and reliable method to measure infection level. But this requires fresh feces, ideally directly from rectum, and McMaster slides, special slide, and microscope. And may not be easiest way for everybody. But let's see what we can see here. This under microscope we can see this kind of eggs, right? So I mentioned a little bit earlier. So this nematogeos or thread neck worm has huge egg, big egg under microscope. So you can recognize this one. And Coccidia oocyst is very small. So you may identify these. But these all the gene worm eggs, they are pretty similar in size. Then you probably not, you will not differentiate one to the other. So like it's even veterinarian, it's difficult. Normally what we do is we culture them, then after they hatch and they become worm, then we can identify which worms are. But when they are egg, it's almost impossible to distinguish or differentiate. This is Barbapol Worm, this is black squirrel worm. 5-point check. So this is not only for worm problem, but general health condition check. Number 1 is nose, number 2 eye, number 3 jaw, number 4 back, number 5 tail. So what do you want to see is number 1, nose. not specific to parasite, but probably respiratory diseases, right? Running nose or something. And number 2, I. This I indicates anemic level using Famacha system. Here we go. This is the purpose of for somebody, some people being here, right? So using Famacha system, you can identify the anemia level of anemia. So this is useful. And JAW, number 3, JAW, bottle jaw or edema is the symptom of hypoproteinemia. And BAC is like body condition score. You can see the how skinny or conditioned the animal's body. Number 5 is tail. That shows you diarrhea or DAC score. So as I said, in natural setting, there is not very often. It's not very often to be infected only Hemoncos, right? So usually if they are infected with Barbapol blood-sucking worms, then they may be infected other worms too, which causes diarrhea, right? So diarrhea can be from other species of internal parasite or gene worms. The next one is packed cell volume, PCV, or for some people, it's familiar, like hematocrit is almost the same thing. Hematocrit is familiar. This is diagnosis of anemia as an indicator of hemoncosis. So how you do it is you collect the blood and put the blood in capillary tube, like very small, tiny, thin tube, and you centrifuge. Then plasma and red blood cell is separated, right? Then this volume of red blood cells or blood cells are more than 30%, then we say normal. But if it's below 20%, then it indicates anemia. It indicates anemia. But this one also requires blood samples and centrifuging. That may not be easy for producers. This is more used for in veterinary offices. Now, Famacha score. So this Famacha score is an indicator can indicate of hemoncosis. Famacha system is developed for sheep in South Africa and find anemia by comparing color of mucous membrane of lower eyelid with Famacha card. The most advantage of this system is do not require special equipment. Everybody can do it with using that card. Only that card is required. However, to use a Famacha card, you must take a Famacha training course and become certified. So here we are. That's the reason why we are here. So later hours, Dr. Steve Hart will explain Famacha system more, but I hope you enjoy this hour to understand the basics of internal parasite. So I will see you later.